In this video, I will discuss the last of the three main components of the cytoskeleton, the actin filaments. So the actin filaments are also known as the microfilaments because they have the least diameter among the three, even smaller than the intermediate filaments. So they function mainly in structural support. Then they function in cell motility, such as in amoeboid movements of certain types of cells like white blood cells, and even in some unicellular eukaryotes. They also help cells, certain cells, to do phagocytosis. And they promote the formation of cleavage furrow in animal cells during cytokinesis in cell division. And just like in microtubules, they also help in intracellular movement of vesicles. And they provide us the primary pathway or railway for vesicle transport in plant cells. Their motor protein, just like that of the motor proteins in microtubules, are actually known as myosin. So in microtubules, those are kinesins and dyneins. So here in actin, the motor protein is called myosin. So this is a monomer of actin. So this is an actin monomer. You can see already from this illustration that the monomer has two ends. There's a positive end, a plus end, and a minus end. And then there is an ATP component, which can then be hydrolyzed by ATPase, so that several of these monomers of actin can actually polymerize together. So if you can remember, in microtubules, it's actually a GTPase driven reaction. But here in actins, it's an ATPase driven polymerization. So when these polymerize, the final actin molecule will have a plus end. That's where new actin monomers are added the fastest. And then we have the minus end. And this is where the removal of actin monomers is fastest as opposed to the addition of actin monomers. So here's an electron micrograph of an actin filament. If we're going to look at this closely, we can see that the filament is actually not just a straight line, but it's composed of these sort of arrowheads that are attached to each other. So we have a pointed end here, and then a barbed end. Okay, so it really looks like an arrowhead. There's a pointed end and then the barbs on one end. So the barbed portion are the plus ends. Okay, that's where new actin filaments are added the most. And the pointed ends are the minus ends. And this is where the removal of new actin monomers or polymers, or components happen. So how do we know that? Because there is this experiment in vitro under glass wherein the scientists have this segment of actin, and they provided this Petri plate that we have here with the monomers of actin, so actin monomers. And so a reaction happens, and we can see here that it's in the plus end where the elongation is greatest, but there is also some sort of extension here in the minus end, but very minimal. Okay, so this is quite different from how microtubules extend in the plus end, because microtubules strictly just extend in the plus end, and nothing really happens in the minus end in microtubules. But here in actin filaments, we can see that there is actually a bit of extension as well, even in the minus end. So here's a more detailed look of how that happens. So we have here a minus end and a plus end of an actin filament. If we put this in a Petri plate with high concentration of actin monomers, what's going to happen is the polymerization of new actin in the positive and negative end will be more or less equal in rate. Okay, look at the arrows pointing towards the minus and the plus end. They are of the same length. That's because the polymerization rate is equal. Where they differ, however, is on the depolymerization rate. We can see here in the plus end that the depolymerization rate is almost zero, while here in the negative end, there is a substantial amount of depolymerization so that the growth of new actin filaments in the minus end is not that fast. Okay, so remember there's still growth here but not that fast. As the concentration of your actin monomers decrease in your Petri plate, for example, the depolymerization rate here in the minus end equals that of its polymerization rate already. So meaning to say there is no obvious growth here. However, in the plus end, the polymerization rate continues to be faster than the depolymerization rate. Therefore, this one grows still. And as the concentration drops some more, notice what happens in the minus end. The depolymerization overtakes the polymerization, and therefore, this portion seems to even shorten. While in the positive end, the polymerization is still faster than the depolymerization. So it still continues growing in the positive end, even if the concentration is now low. Until such a time that the concentration of your actin monomers in your solution remains constant. Why? Because the polymerization in the positive end, look at the arrow here, is balanced with the depolymerization in the negative end. So the number of new actin monomers coming in the positive end equals the number of actin monomers that are leaving in the negative end. So there's as if no growth in your new actin filament here. So this process is actually reminiscent to scientists of a treadmill, right? Okay, it's like the platform of a treadmill moving from right going to the left. Okay, so that's why this model of elongation of actin is known as treadmilling. So I hope you can compare this with the polymerization of microtubules. Okay, if you go back to the model there, which is the catastrophic model. Right, so now let's talk about the motor proteins of actin, and these are the myosin. And they resemble the basic structure of the kinesins and the dyneins from the microtubules, but myosins are relatively larger molecules. They are also being powered by ATP. So this is how they look like. They look like mango sprouts here. Okay, we can see the head and then the tail. And of course, this one is myosin, and there are many types of myosins. The first type we call myosin type 2 is the conventional myosin. It is the one found in muscles. Okay, the muscle myosin. And these types of myosin assemble themselves into bipolar filaments. So instead of just being one molecule like this, they actually group themselves back to back like this one, this bipolar filament. So this is the natural state of myosin 2. Okay, they're bonded together with other myosin 2s in a back-to-back manner. And we can see here a picture of that. So here are the heads, and then here are the tails in the middle bundled together. And then we have unconventional myosins like myosin 1. So myosin 1 provides us a cross-link between actin in the cytoskeleton and the plasma membrane. It is unable to assemble into filaments, and it is single-headed, so as we can see here. So simply as cross-links between actin in the cytoskeleton and the plasma membrane. So it anchors this actin over here to the plasma membrane, so that whatever movement the actin will do, the plasma membrane may also sort of change in shape. And then we have myosin 5. So this myosin is similar in function as kinesins and dyneins because they carry vesicles along, this time, actin filaments instead of microtubules. So they move in a minus to plus direction. So they are unable to form into filaments. They are double-headed with a neck consisting of 6 light chains, so fairly long, as you can see here. So they also, it looks like long legs, as you see here. So we have the head here, but then the neck is very long. And then at this portion of the tail, they carry the vesicle that they are going to move about from a negative to positive end of the actin filament. And because they have long legs here, they can also move considerably faster than, say, kinesins. And then the equivalent of the dyneins would be the myosin 6, which moves from plus to minus direction in actin filaments. So here we can see a picture of a fast-moving myosin 5 molecule. You can see here the sort of inverted V shape. So here's the myosin 5. You can just see the tail, and look how the position changes with respect to the change in time. So it's moving. So this is a diagram of that. So you can see here the myosin 5. If ever there are certain obstacles along the way as it moves along the actin filament, the myosin 5 or 6 can actually lift one of its neck and head to just go through the obstacle, and it can continue moving. So imagine someone, a runner that's trying to go through an obstacle. Instead of hitting the obstacle, it just lifts one of its legs so that it can pass through. So in this illustration, we can see how one vesicle, as shown here, may actually be moved by a myosin molecule, as we can see here on top, or in this case, a kinesin molecule, as we can see here in the bottom. So it's quite busy. Both of the microtubules and actin filaments are used as railways by dyneins, kinesins, and myosins in moving vesicles and organelles inside the cell. So one very important role of myosins and actins, of course, is in the movements of muscles. And they are quite obvious in terms of appearance, the banded appearance in skeletal muscles, for example. So here we can see a muscle fiber, which is actually a muscle cell composed of myofibrils, which in themselves are striped, and the stripe pattern is actually the sarcomere of that myofibril. Okay, and the sarcomere consists of these thick lines in the center, which are the myosin, and then these thinner lines on the sides, which are the actin. So here's another view of a skeletal muscle. So we call this striated muscles because of the striations. Well, I think this is not skeletal. It looks like it's cardiac because of the interconnections between the muscle fibers here. But anyway, both cardiac and skeletal muscles are striated. okay, as opposed to smooth muscles. So here's another illustration. So we have these terms. We have the A bands, the H bands, and then we have this sarcomere. This is one unit of the sarcomere, and the boundary would be the Z line, the boundary between the two sides of the sarcomere. So here's a closer and a clearer view. We have the Z lines on both ends, and then we have the actin filaments. Here's the positive end. Here's a negative end of the actin filament. There's also another one here. And here in the middle, we can see the myosins, the muscular myosins. So these are the typical myosins, and they are bipolarly bonded to other myosins. Okay? And these are their heads that are protruding, and then these are their tails in the middle. So when the muscle is relaxed, the connection or the cross bridge between the myosins and the actins is absent. And so the muscle is its usual length. But when the muscle contracts, the myosins heads actually are now in contact with the actins, and they sort of pull the two actin components from each side of the sarcomere towards the center, and the whole muscle fiber shortens or contracts. Well, there are other muscle proteins, as we see here. We have the ones that keep the myosin in line here in the middle between the two actins. We call this the titin. And then we have the boundaries between these actin myosin components, the nebulins, the green proteins. And then there's even another protein or other proteins that are found wrapping around the actins here. We have the tropomyosin, this elongated strand that we see here, and then troponin at certain intervals along the actin length. So with the help of ATP, of course, it's still ATP that powers myosin, your muscle is able to contract, as shown in the sequence here. So your muscle is only able to contract when, especially if we're talking about skeletal muscles, when nerve signal from a motor neuron dictates it to contract. So a motor neuron plus all of the muscle fibers that it innervates is known as a motor unit. Okay, so the green motor unit here only powers these certain muscle fibers that are innervated by the neurons. And the blue-colored motor neuron or motor unit here only innervates these other muscle fibers. So here's a closer look. So when a nerve signal reaches a muscle cell or a muscle fiber, it will actually travel through the plasma membrane and through this transverse tubule of the muscle cell. And as it travels through this transverse tubule, it's going to stimulate the sarcoplasmic reticulum, which is actually a special type of endoplasmic reticulum that stores calcium ions in the muscle fiber to release calcium into the cytoplasm. And this release of calcium allows the actin and myosin units in the muscle fiber to actually contract. So how does that happen? The calcium will actually bind to the troponin, okay, this protein. So remember, these beads here are the actin, okay, and then this long molecule here is a tropomyosin. Without calcium ions, the tropomyosin is covering the myosin binding site of the actin. So those circles found in the beads. But with the presence of calcium, calcium will bind to the troponin, and the troponin will cause the movement of the tropomyosin, allowing the exposure of these circular-shaped myosin binding site. And once they are exposed, myosin can then bind to them and do its work to contract the muscle fiber. So here's a look on that. So we have the nerve signal travels through the plasma membrane into the transverse tubule, causes the release of calcium ions out of the sarcoplasmic reticulum, causes the binding of those calcium ions onto the troponin, thus moving the tropomyosin and exposing the myosin binding sites of the actin. And the myosin will be able to bind to the actin, and with the help of ATP will do a power stroke, and therefore the muscle contracts. And here are some other examples of actin binding proteins, sort of similar in function as the microtubule associated proteins.